Welcome back. This is the fourth and last chapter of lecture 2. I will talk about EEG, electroencephalogram, and EMG, electromyogram signals. Let's see first the EEG signals. In your brain, the most prominent cells are the so-called pyramidal cells. So you have 2/3 of all cells from these type of cells, the pyramidal cells. Here you can see dendrites of the cell. This is the income, so signal is coming through these dendrites, and it is going through the axon, which you can see here. But it gets its name from the shape of its cell body. This is the pyramid which you have here. So these are the main cells of your brain for doing all the signalings, all the connections between your brain and your body. Here you can see that the signal is coming in from here, so the depolarization starts there and it will go out here. So if you would like to draw again those circles of electricity, it will start here and close on the axons. And here you can see that from a slice of a brain, this is how all these cells are arranged in this slice, your cortex, the dark purple color over here, and you can see that these pyramidal cells are always standing in upside. So if you have this cortex, it will be always orthogonal to the surface of the cortex. Here it will stand like this, but if you go here, it will stand like this to be orthogonal to this cortex. So if you are measuring these circles from here, this is the gyrus, then you will have these loops closing like this. While if you are here down in this curve, then you will have the loops closing like this. And if you are putting the electrodes onto the surface of the skull, then you will have signals nicely measurable from these gyri, but you cannot really measure the signals coming from here, as these loops will close here and you will not be able to measure these circles with the surface electrodes. So we are usually measuring signals with the surface electrodes from these parts of the brain, of the cortex. How do we do this? So this is the standard EEG measurement positions. This is the European International 10-20 system, where you have all these electrodes placed on your skull, and you have these 10 and 20 numbers as percentages of distance between the electrodes. So how to place them, where to place them spatially, this is what these numbers are telling you. And you can see the measured signals from there in this diagram. You will need a ground, which is usually the earlobe. So you put one electrode here and you will measure the difference between this earlobe and one of these many electrodes. And you can see all these names for the electrodes, like P1, C, and so on. This is what you will see in an EEG measurement. So if you download the signal like this, then you will know where this measurement was taken, as they always put the electrodes like this in this system. But what can you measure? So you are far away from the cells themselves. So you will measure surface potentials on the skull, and the bone is attenuating a lot from these signals. So these will be very low voltages. You will need very good electrodes to measure these signals. But what can we see? So we usually can observe some different oscillations, like the alpha wave. You must have heard about that when you are relaxing, then you can measure the alpha wave. But there are many others. So we usually talk about the alpha, beta, gamma, delta, and theta waves. And this alpha wave which is coming from a restful awake state. Delta and theta waves are coming from a sleep state. Usually delta signals deep sleep, or it can be measured from infants or from serious brain damaged persons. Then the theta wave, apart from as a sleeping signal during one stage of your sleep, can be measured in children and when someone is very frustrated or has very prominent emotions. Then you can have the beta wave while you are thinking. So when you are thinking a lot and your eyes are not closed, you are not relaxed, this is the waveform which will be measured. And the gamma wave is usually measured when you are linking together some perceptions and you are thinking through many different perceptions. So you are seeing something and listening to something and reading something, then you usually can see the gamma waves, but it's not proven yet. And then where can we measure all these signals? So in this diagram you can see where these signals can be measured. The first one we have talked about was the alpha wave, which is during a relaxed state when you have your eyes closed. This signal can be measured from your occipital lobe, which is in the back of your head here. This frequency range is between 8 and 13 Hertz usually. Then you have a very similar signal, which we haven't discussed before. This is the so-called mu wave, which has the exact same frequency range between 8 and 13, but is connected with movement. You can measure it from your parietal lobe up here, as you can see it here, which is during thinking about some motor movement or actually performing some motor movement, and also has this frequency range of 8 to 13 Hertz. The other wave we have discussed was the delta wave, which was during deep sleep, and this is usually between half a Hertz and 4 to 5 Hertz, and it can be measured on your frontal lobe over here. You can measure the beta activity here, which was the thinking, on your frontal lobe, which was above the 13 Hertz, so it's somewhere between 13 and 30 Hertz. Then you can measure the gamma wave, which was this perception fusing all the different information, also on your parietal lobe. This is this gamma wave, which is about 30 Hertz usually. So it's a very high frequency. This is the highest frequency and hence the least measurable frequency, because this is the one which will be attenuated the most because of all this tissue, the bone and the skin and the hair on your scalp. Yes, and then you have the theta activity, which is, as we have discussed, in children or also during sleep. So there is a sleep stage where you can measure it, or when you are very tired, drowsy. And this can be measured from your hippocampal rhythm. So it's present in your cerebellum, your small brain, and the hippocampus, which is over here behind all these structures. So these are the different waves you can measure from the brain, and you can give some information about the activity of the brain. This is what we use them for. So you can tell which sleep stage the person is in. You can see an epileptic seizure, which will be a very prominent change in all these brain waves. But you can track whether somebody is thinking or is relaxed and not listening to the lecture, and so on. So the next one will be the EMG signal, the electromyography, in which case we are measuring signals from our muscles, how they are contracting and how they are moving. The first term we have to discuss is this motor unit axial potential. So what is a motor unit? In a motor unit you have one neuron, which is controlling the muscle, and you have many muscle fibers. So one neuron is connected to more muscle fibers and will activate them to contract. This will be one motor unit, and you can measure some potential from this motor unit. But as we had with the different parts of the heart, they will be from different directions, different distances. So as all these waveforms are adding up, you will have something different than one action potential. And this is the usual waveform you can measure from a muscle motor unit. It is biphasic, so you have downgoing and upgoing compared to the resting state of it. And it is usually between 0.25 and 5 millivolts, and you can diagnose different problems of the muscle by measuring this signal. So, what is happening when you need more force? So you are holding this pointer, but then you will grab it and squeeze it more and more and more. So first you need only some of your muscles, but then you need more muscle strength. You can do two different things. One is the spatial recruitment and the other is the temporal. So in one, in the spatial recruitment, you will use more of your muscle fibers. You will activate more motor units. And in the other one, in the temporal recruitment, you use the same motor unit, the same fibers, but with an increasing frequency. So you will fire these contractions more and more to have this increased force, muscle force. In this diagram, you can see both of them. So here you can see that the new waveform is arising, which is from a new motor unit because it has a totally different shape, so it has to come from somewhere else. But you can also see that instead of two of these waveforms, now you have three within the same duration. So you also have temporal recruitment, not only spatial. And here you see that there are even more different waveforms. So you have a third waveform, like here, but you have from all of them more. So you have spatial and temporal recruitment here. And what can we measure on the surface? So on the surface, you cannot separate only one motor unit. You will have the influence from all the surrounding motor units, and these will add up. So you will have to add up all these different waveforms, and you will have something very new, very different, very chaotic. So while here you see the motor units, when you add up all of them, you will have some oscillation, some change. It's not even periodic. But then what can we measure? So if you're in a relaxed state, you won't have only noise here, so small unintentional contractions of the muscle. But if you are squeezing something, then this will jump. And this is what we are usually looking for. So if you would like to control prosthesis, a prosthetic, then it is looking for this change in the muscle signal, and when you have this abrupt change, so the amplitude will grow, you know that, okay, now I have to control the prosthetic and it has to close the hand, for example. Yes, and here you can see what happens in neuropathy and myopathy. You have very different waveforms from a normal signal. So if you have neuropathy, you will try to use the same muscle motor units, muscle fibers, with an increased frequency, but the whole waveform will be just amplified and it's not really working. While in myopathy, when not the neuron but the muscle fibers are themselves damaged, then it will recruit other, so you will have spatial recruitment of other motor units, which you can see here. And this is an example of increasing muscle force when you are in a resting state and then you start to squeeze more and more like your wrist and it will grow up. So this is something you can measure. Or in walking, when you are standing on one hand and then you start to step. So this is another example of, okay, here, so now I have to control the other leg, so, the prosthetic on the other leg, for example. So these are examples of how to use these measurements. You can use them for controlling and you can use them for diagnosing different pathologies of the muscle or the neurons in your body. So thank you. This was the second lecture of biomedical signal processing. We have discussed how mechanical and optical signals can be measured from our heart, like the blood pressure, like the phonocardiogram, and these optical signals which can be measured with LEDs, the PPG and the PO signals. Then we have seen how electricity is generated in our body, so how one cell is generating the electricity, what is electroneurography. And then we have seen what is electrocardiography, so electric signals from our heart. And then we have discussed how signals from our brain and from our muscles are emerging in the body. Thank you very much.